Greetings from the Department of Botany, St. Xavier's College, Palayamkottai. I am Dr.Joelri Assistant Professor in the Department of Botany, specialized in molecular systematics. So I am dealing with the subject Plant Anatomy and Embryology for undergraduate students, under which I am going to detail explaining about what is anomalous secondary growth. Now, what do you mean by anomaly? Anomaly is something that is deviated from the normal. So if there is an anomalous secondary growth, there should be a term called normal secondary. If there is a secondary growth, definitely there should be a primary growth. Growth in plants is very interesting and fascinating. Let us go into this fascinating topic of anomalous secondary growth and understand the details and the ecological significance behind it. Now, understanding the basics of secondary growth. Secondary growth is a process through which many plants increase their stem or root diameter. Now, what is diameter? That is increasing in the girth of the stem. This growth occurs in woody or perennial plants, in contrast to the primary growth seen in all plants. That's why I told you to understand the concept of primary growth, normal secondary growth, or anomalous secondary growth. While primary growth adds to the length, the plant or the tree grows in height, whereas secondary growth results in increase in the girth or thickening and strengthening of the stem for its support and transportation efficiency. Now, this is normal secondary growth that happens in most of the woody perennials. Now, coming to anomalous secondary growth, it means anomalous thickening that occurs in herbaceous plants and non-woody perennials or sometimes in biennials, exhibiting traits of typically woody plants. It is an extraordinary adaptation, you can say an ecological adaptation, that allows these plants to increase in the diameter and adopt the characteristics of woody plants, even though they are not classified as such. For example, Boerhaavia is a herbaceous plant and it is a perennial, but it is adapted to anomalous secondary growth. Now, this feature of plants adapting to anomalous secondary growth is fascinating and has a lot of impacts in the ecology. Let us understand the depth of anomalous secondary growth. Now, let us see the different tissues involved in anomalous secondary growth. Vascular cambium is normally present in all the dicots as it exhibits normal secondary growth. We can also see the cork cambium in the normal secondary growth of dicots. But the presence of cork cambium in monocots is a peculiar feature. The vascular cambium, unlike the classical secondary growth, anomalous secondary growth in certain plants does not rely on the vascular cambium, which is a very important point to be noted. The anomalous secondary growth not relies on the vascular cambium, whereas it relies on special type of cambium that is formed during the anomalous secondary growth. For example, the normal ring that is formed in the dicot plant is accompanied by certain extravascular bundles in the pith region. Normally, we don't have vascular bundles in the pith region, but there are certain cells that has started to divide and form cambial cells in the pith region to form medullary vascular bundles. You see, the cork cambium is actually present in the cortex region, where the vascular cambium is not the primary driver, Cambium starts to play the role in anomalous secondary growth, where it contributes to thickening of the stem orbit. Apart from the vascular cambium and the card cambium, there are secondary meristers that play a key role in anomalous secondary growth, typically found in specific plants. And these meristers, such as intravascular cambium, undergo proliferation to facilitate the anomalous thickening. Now, here you have to understand that in the normal cambium, there are two types. One is the interfascicular cambium, the other one is the intravascular cambium. Interfascicular cambium is present in between two vascular bundles, whereas intravascular cambium is present in between the xylem and phloem in one vascular bundle. Now, here the intravascular cambium undergoes proliferation and also facilitates in anomalous thickening. Let us go into the details of anomalous secondary growth. It is an abnormal type of secondary growth. As I already told you, it is an anomaly. As we have seen, the secondary growth is normally present in dicots, whereas there is no normal secondary growth in monocots. For example, in Dracaena, Yucca, and agave, people are fascinated about how these plants started to increase in thickness, especially the stem feature to be noted. In all the arbors and monocots, the secondary cambium starts to grow in the hypodermal region. What do you mean by hypodermal region? Hypo means below, and dermal means epidermis. The cells or tissues below the epidermis are called as the hypodermal region. And the functioning of the secondary cambium or the car cambium in the hypodermal region is an important indication of the presence of anomalous secondary growth in monocot plants. Now, this secondary cambium forms conjunctive tissue and patches of meristematic cells. The meristematic patches grow into secondary vascular bundles. Let us understand some more details of what is anomalous secondary growth. Where anomalous vascular bundles also start to occur in cortex. You see, in the case of dicots, we see the normal secondary growth. How to identify the anomalous secondary growth in dicots? In dicot plants, we can see the car cambium in the cortex region, or otherwise the hypodermal region. We can also see the normal thickening. How to identify anomalous secondary growth in dicots? The important indicating feature of anomalous secondary growth in dicots is the presence of vascular bundles in the cortex, called as cortical vascular bundles present in nyctanthus. Sometimes these anomalous vascular bundles also occur in the pith region, for example, Boerhaavia. Now, these are all special indications for anomaly in dicot stem. Very obviously, the storage roots of beetroot. Not only stem undergoes secondary thickening or anomalous secondary growth, even the roots undergo anomalous secondary growth by the accessory cambial rings that is found outside the epidermis. They produce less secondary xylem but more secondary phloem. The secondary phloem contains abundant storage parenchyma. Now, these points contribute to the anomalous secondary growth. Now, coming back to the monocotyledons, most of the monocot plants belong to the family Liliaceae. I already told you Dracaena, Yucca, cordyline, agave, aloe. All these plants exhibit a peculiar type of secondary increase in thickness. See, another important feature is that these plants are used in indoor gardening and other medicinal properties also are found in these plants. Dracaena, Yucca are normal ornamental plants found in gardens, outdoor gardening as well as indoor gardening. Cordyline, agave, aloe are well-known ornamental species. Now, the study on anomalous secondary growth in such species will be interesting and help us to grow such plants with different ecological adaptations. Now, the depth of anomalous secondary growth can be divided into two types. That is anomaly observed in the cortex region and anomaly observed in the vascular bundle region or the vascular tissue region. What is the abnormality observed in the cortex region? See, the regions which have ceased to elongate, for example, mostly the cortex cells are well differentiated, especially the cortex cells have the chlorenchyma cells, collenchyma cells in Dicot plants, and spirenchyma metastasis in Monocot stem. Now, all these cells are well differentiated. Now, these differentiated cells revert back to the meristematic state, forming a special type of kidney. That is abnormality that is noted, especially in the Dracaena species, which we are going to deal specifically in this video lecture. The cortex cells becomes meristematic, which is an important indication for anomaly. The secondary meristem originates in the cortex. In fact, deep layers of cortex are very cyclic. Now, there are still more studies, and it is very difficult to locate these zones in distance. See, we do not know the genetic mechanism behind it, and research on genetic mechanism behind the redifferentiation, and the differentiated cells reverting back to the meristematic state will prove improving results or very promising results for tissue culture. Abnormal vascular system. See, in abnormal vascular system, we can find out the cambial cells of fusiform type and radial type. Fusiform type cambial cells look like they have tapering ends. Now, let me show you using a pen to differentiate the fusiform cambial cells. See, in abnormal vascular bundles, you can see certain cells with tapering ends. Now, I am drawing it here. Now, here, now these two ends are tapering. You have the nucleus in the center. Tapering ends. Now, such type of cells are called fusiform cells. Sometimes cell may be rectangular, which are called rectangular cambial cells. In abnormal vascular system, you can see these fusiform and rectangular cambial emissions. Instead of forming phloem and xylem on the outer and inner side as in normal condition, where phloem is formed towards the periphery and xylem is formed towards the center, the cambial cells go on dividing and producing secondary tissues on the inner side first. And later, small amount of new tissues are cut off on the outer side as well. Another important abnormal condition that is observed in the vascular system is those found on the inner side differentiate into oval-shaped vascular bundles. And radially arranged parenchyma cells are also found. Now, these parenchyma cells in which vascular bundles remain embedded are said to constitute the conjunctive tissue. Now, parenchyma cells in the vascular bundle picture constitute the conjunctive tissue. Now, this is found in normal growth also, normal secondary growth also. A few derivatives of the cambium divide longitudinally, initially, anticlinally, then periclinally, and even horizontally to form xylem and phloem elements of secondary vascular. So, what do you mean by this? There are different types of divisions. One is anticlinal, and other is periclinal divisions. Now, let me explain you what we mean by anticlinal divisions. Take, for example, this is a cell, a plant cell. If this plant cell undergoes vertical divisions on this plane, this type of division is called anticlinal divisions. If the same cell undergoes horizontal divisions, can you see here? Now, this type of division is called as periclinal division. So two types of division, anticlinal and periclinal. What will be opposite divisions? There is no type of division that can be explained where anticlinal, periclinal, and divisions on different planes occur. Opposite division means cell divides in any plane. It can divide in this plane, or vertically, or horizontally. These type of divisions is called opposite divisions from xylem and phloem elements, and this is one of the important indicators of abnormal vascular system. Now, these differ from the primary ones, where in the presence of small amount of phloem and in the absence of annular and spiral protoxylem elements. Now, all these are basic indications of abnormal vascular system. Either it be monocots or dicots. The secondary bundles are mostly amphivasal. What do you mean by amphivasal vascular bundles? Let me explain you what is amphivasal vascular bundles. We'll also see in detail about these amphivasal vascular bundles with a typical example of Dracaena. Now, amphivasal vascular bundles, a special type of vascular bundles, where xylem is on the outer side, whereas phloem is on the inner side. See, normally, in primitive plants like pteridophytes, we can observe the xylem in the center. I will mark it like this. Xylem can be marked like this, so that you clearly understand what is xylem and what is phloem. The xylem is in the periphery, and phloem is in the center. Whereas in primitive vascular bundles, xylem is in the center, whereas phloem is outside the xylem. Now, this type of vascular bundle where phloem is outside and xylem is inside is called as amphicribal vascular bundles. Amphicribal vascular bundles. I am trying to write it here so you understand what is amphicribal vascular bundles. So, amphicribal, CRIBAL, amphicribal vascular bundle. Since I am using the PowerPoint pen, it may not be very clear, but just try to understand that it is cribal, CRIBAL, amphicribal vascular bundles, where xylem is in the center and phloem is towards the periphery. Here, phloem is the center and xylem is in the periphery. Now, this type of vascular bundles are advanced type of vascular bundles that are found in monocot elements. Now, this means the evolutionary significance where amphicribal vascular bundles are turning into amphivasal vascular bundles that can be noted in the anomalous secondary growth of monocots, and especially in some arborescent monocotyledons. Normally, the secondary bundles or the vascular bundles are collateral. Now, what do you mean by collateral? Collateral is a condition where xylem and phloem are arranged in the same radius. And conjoined is a condition where xylem is towards the central region or the pith region and phloem towards the periphery. Now, this is a normal type of vascular bundle found in a normal dicot stem. So, collateral vascular bundles are found both in normal vascular bundle system as well as normal vascular system. But what is significant is the presence of amphivasal vascular bundles, especially in arborescent monocots like Dracaena. The small amount of phloem consists of short sleeve tubes, which can be observed in a higher magnification, and companion cells and some parenchyma. Xylem is made of only tracheates. It's another important indication of abnormal vascular bundle. There are no other elements of xylem like xylem vessels or xylem parenchyma or xylem fibers. Xylem is made up of only tracheates, and they form scalariform thickening, which is otherwise called as the ladder-like thickening. So we see the ladder. So thickenings are in the form of ladder. Now, if thickenings are in the form of ladder like this, now such type of thickenings are called as scalariform thickening. and tracheates undergo only scalariform thickening in abnormal vascular system. The tissues cut off with the cambial cells on the outer side are scanty in amount and the parenchymatous in nature, and the primary bundles are comparatively larger. They are also mostly amphibasal and rarely collateral. So, as I told you, amphiβasal vascular bundles are very important indication for abnormal vascular system or anomalous secondary growth. Let us take up the example of a typical monocot stem TS of Dracaena showing anomalous secondary growth. So these two pictures are the transverse section of the Dracaena stem, and you can see this particular portion where the xylem which is more stained. Normally xylem has thick cell walls and are well stained by safranin, whereas phloem cells have only primary cell wall and they are not much stained by the safranin. So safranin is a stain that is used to stain the xylem and the phloem. Now it's different here where xylem cells are stained more and phloem cells are not stained by saffron. This is because of the chemical composition of the cell wall. Now we can see xylem outside and phloem towards the center. What is this vascular bundle? It is amphibasal vascular bundle. Now this indication of the presence of amphibasal vascular bundle is more than enough to identify that this particular stem has undergone anomalous secondary growth. Not only the presence of amphibasal vascular bundles, the presence of a large cortex. Normally, the cortex region is very short in monocots, but the length of the cortex in the Dracaena stem is very large. What is the reason? And this is contributing to the thickening of the stem. This is because of the abnormal activity of the cork cambium, which starts dividing, not forming xylem and phloem, whereas the secondary tissues increasing in the girth of the stem. Moreover, we can also see the cortex cells reverting back to the meristematic state and starting to function as cambium. Let us see some more details of the Dracaena stem TS so that you understand the anomalous secondary growth in Dracaena much better. Now this picture is the outline drawing of the Dracaena stem TS, what we have observed in the previous slide. Now this is system which is a woody perennial. Now how this woody nature comes to a monocot is because of the anomalous secondary growth. Now you can see the ruptured epidermis followed by the cork cells. Now these cork cells are because of the activity of the cork cambium. The cork cambium divides to form cork cells towards the periphery and secondary cortex towards the center. Now this also contributes to the anomalous secondary growth in monocots. And you have the normal cambium that is not present in the monocot liddens. So what is this cambium? The presence of cambium itself is an anomalous condition in monocots. This cambium starts dividing, not in the normal form of forming xylem towards the center and phloem towards the periphery. Here, both the xylem and phloem are formed towards the center, and this xylem surrounds the phloem. So this type of vascular bundle is called as amphibasal vascular bundles. You can see the indication of phloem, the center, and xylem towards the periphery. And you can also see the presence of conjunctive tissue because of the division of the cambium. And you can see the pith region scattered with numerous vascular bundles, which are specially called as amphibasal vascular bundles, which is a special indication of anomalous secondary growth in Dracaena. Let us see another important picture of anomalous secondary growth, where you can very clearly see the presence of cork cells and the formation of the secondary cortex and the numerous amphibasal vascular bundles in the center. All these sections are taken in our own lab so that you are fascinated and get interested in these topics. Now look at another outline of the section of the TS of Dracaena showing amphibial vascular bundles. I am showing you this picture so that you get more clarity on the presence of the amphibial vascular bundles, which is one of the more important features of anomalous secondary growth. We can also see the primary tissues and the secondary tissues, and the cork cells, and the cortex that is formed, and the large cortex that is formed. Now, in this picture, we can see the presence of lenticels. Now, what are lenticels? Now, they are advanced features of the stomata. These stomatal openings become lenticels during the anomalous secondary growth. Now, these lenticels are very important for the gaseous exchange, as and how stomata is used for the gaseous exchange. Once the arborescent monocot starts becoming bigger and bigger in thickness, these lenticels are formed and help in the gaseous exchange. And below which you have the cork cells, the cork cambium, or the abnormal cambium that is formed, which contributes to the anomalous secondary growth. And here, the double-lined portion, which clearly indicates the xylem towards the periphery or towards the outer part of the vascular bundle, and phloem in the center. Now, all these pictures are for better understanding, which you can go through, will be uploaded in quadrant 2. Now, let us see the anomalous secondary growth in dicots. So far, we have seen the presence of anomalous secondary growth in monocots is a very important ecological adaptation, so that they get adapted to the woody perennial condition, so that they can survive in an environment for a longer period of time. So what about the Boerhaavia stem? This is a typical example for a dicot stem showing anomalous secondary growth. Finding out an anomalous secondary growth in monocots is more easier. How to identify anomalous secondary growth in dicots? See, this picture or the transverse section taken in our lab shows the presence of normal vascular cambium forming a ring-like structure. You can see the ring that is formed. So you can see the ring that is formed here. Now this is the normal vascular bundle that is present in all the dicots. What is the anomalous secondary growth pressure? Apart from the normal ring of vascular bundles, there are certain abnormal vascular bundles found in the pith region. Normally, vascular bundles are not found in the pith region. Here, in the TS of stem of Boerhaavia, you can observe abnormal vascular bundles in the pith region, which are called as medullary vascular bundles. And you can observe two large vascular bundles and other smaller vascular bundles around. The presence of medullary vascular bundles is by itself an important feature showing the presence of anomalous secondary growth in dicots. So you should be able to identify what is normal secondary growth and anomalous secondary growth, especially in dicots, where dicots undergo both normal secondary growth. Some of the dicot plants undergo both normal secondary growth as well as anomalous secondary growth. And Boerhaavia is one of the typical examples of anomalous secondary growth. Now let us go very deep into the next slide to understand the anatomical details of Boerhaavia stem. Now here we can see the presence of xylem, which are darkly stained, and conjunctive tissue, and the cortex cells start dividing, and cork cambium is formed in dicots also. You can see even the normal secondary growth, the presence of cork cambium, where the cork cambium divides on the outer to form the cork cells. or otherwise you can call it as phellum. The parenchyma is otherwise called as phellogen. This parenchyma of phellogen divides towards the center, forming the secondary cortex or phelloderm, phellum or cork cells, phellogen or parenchyma, phelloderm or the secondary cortex. Three layers are formed in the outer region below the epidermis, the hypodermal region, which also contributes to the increase in the depth of the Boerhaavia stem. Apart from which, there are also presence of medullary vascular bundles. So this picture shows the ground plan. This is the ground plan of the TS of the Boerhaavia stem, showing two large medullary vascular bundles apart from the normal ring of cambium, where you can see cambium in between two vascular bundles, which is called as the interfascicular, INTR, interfascicular vascular bundles. Whereas the cambium that is present within the vascular bundle is called as intrafascicular vascular bundles. So, interfascicular vascular bundles, writing it in short, FVB, intrafascicular vascular bundles. Together merge and form the normal ring. So, normal secondary growth also occurs in Boerhaavia. Anomalous secondary growth also occurs in Boerhaavia. Both type of secondary growth are occurring in Boerhaavia, contributing to increase in the girth of the stem. It's a very interesting feature. It is a herbaceous perennial, and this herbaceous perennial survives for a longer time because of this anomalous secondary growth. That is ecological significance of anomalous secondary growth. Let us see one more picture of the enlarged portion, where you can see the cambium even in the medullary vascular bundles. Cambium is not only present in the normal vascular bundles, cambium is also present in each medullary vascular bundles, which is also an important feature to be observed in the anomaly of Boerhaavia TS study. Now this picture shows you very clearly about the interfascicular and intrafascicular cambium, where you can see the medullary bundle ring, the large medullary vascular bundle in the center, having a very prominent cambium and cuts the cells of phloem towards the periphery and cuts cells of xylem towards the center. Now it acts in the normal way, not like the vascular bundles in Dracaena, that is monocots. The vascular bundles in monocot stem divides in a very peculiar way, forming amphivasal vascular bundles. Whereas, though the anomaly present in the Boerhaavia stem TS forms the medullary vascular bundles, the medullary vascular bundles also function in a similar way like normal vascular bundles. Now, we have seen the differences between the anomalous secondary growth in Dracaena and the Boerhaavia. In short, we can say the presence of amphivasal vascular bundles by itself is an important indicative feature of anomalous secondary growth in monocots, whereas the presence of medullary vascular bundles is an important feature in anomalous secondary growth of dicots. There are also other many features of anomalous secondary growth observed in different other plants, which can be discussed in the part 2 anomalous secondary growth video. To sum up, let me explain you. We have seen what is anomalous secondary growth or what is anomaly in secondary growth. By finding out the difference between what is primary secondary growth, normal secondary growth, and anomalous secondary growth. Then we have seen the secondary growth in monocots, especially in Dracaena, and also secondary growth or anomalous secondary growth in dicots. Having an example of Boerhaavia. Let us get a deep insight of other types of anomalous secondary growths occurring in different other dicots and monocots in the next video. Thank you one and all for watching this video. Now you could understand the two pictures behind me showing the anomalous secondary growth. One on the right hand top corner and one on the left hand bottom. The left hand bottom picture shows the anomalous secondary growth in monocots, whereas the right hand top picture shows anomalous secondary growth in dicots. Thank you one and all. Let us meet in another fascinating video on anomalous secondary growth in monocots and dicots to get a very deeper insight and more understanding. Thank you.